Monocyte.
Monocytes are a type of white blood cells. They are the largest of all leukocytes. They are part of the innate immune system of vertebrates including all mammals, birds, reptiles, and fish. They are amoeboid in shape, having clear cytoplasm. Monocytes have been shaped nuclei that are unilobar, which makes them one of the types of mononuclear leukocytes. Monocytes constitute 2% to 10% of all leukocytes in the human body. They play multiple roles in immune function. Such roles include replenishing resident macrophages under normal states, and in response to inflammation signals, monocytes can move quickly to sites of infection in the tissues and differentiate into macrophages and dendritic cells to elicit an immune response. Half of them are stored in the spleen. Monocytes are usually identified in stain smears by their large kidney-shaped or notched nucleus. These change into macrophages after entering into the tissue spaces.
Physiology.
Monocytes are produced by the bone marrow from precursors called monoblasts, bipotent cells that differentiated from hematopoietic stem cells. Monocytes circulate in the bloodstream for about 1 to 3 days and then typically move into tissues throughout the body. They constitute between 3 to 8% of the leukocytes in the blood. Half of them are stored as a reserve in the spleen in clusters in the red pulps cords of Billroth. In the tissues, monocytes mature into different types of macrophages at different anatomical locations. Monocytes are the largest corpuscles in the blood. Monocytes which migrate from the bloodstream to other tissues will then differentiate into tissue-resident macrophages or dendritic cells. Macrophages are responsible for protecting tissues from foreign substances, but are also suspected to be important in the formation of important organs like the heart and brain. They are cells that possess a large smooth nucleus, a large area of cytoplasm, and many internal vesicles for processing foreign material. Monocytes and their macrophage and dendritic cell progeny serve 3 main functions in the immune system. These are phagocytosis, antigen presentation, and cytokine production. Phagocytosis is the process of uptake of microbes and particles followed by digestion and destruction of this material. Monocytes can perform phagocytosis using intermediary, opsonizing, proteins such as antibodies or complement that coat the pathogen, as well as by binding to the microbe directly via pattern recognition receptors that recognize pathogens. Monocytes are also capable of killing infected host cells via antibody-dependent cell-mediated cytotoxicity. Vacuolization may be present in a cell that has recently phagocytized foreign matter. Microbial fragments that remain after such digestion can serve as antigens. The fragments can be incorporated into MHC molecules and then traffic to the cell surface of monocytes, and macrophages and dendritic cells. This process is called antigen presentation and it leads to activation of T lymphocytes, which then mount a specific immune response against the antigen. Other microbial products can directly activate monocytes and this leads to production of pro-inflammatory and, with some delay, of anti-inflammatory cytokines. Typical cytokines produced by monocytes are TNF, IL-1, and IL-12.
Monocyte subpopulations.
There are at least 3 types of monocytes in human blood. A. The classical monocyte is characterized by high-level expression of the CD14 cell surface receptor, CD14++ CD16 monocyte. B. The non-classical monocyte shows low-level expression of CD14 and additional co-expression of the CD16 receptor, CD14+ CD16++ monocyte. C. The intermediate monocyte with high-level expression of CD14 and low-level expression of CD16, CD14++ CD16+ monocytes. There appears to be a developmental relationship in that the classical monocytes develop into the intermediate monocytes to then become the non-classical CD14+ CD16++ monocytes. Hence the non-classical monocytes may represent a more mature version. After stimulation with microbial products, the CD14 plus CD16 plus monocytes produce high amounts of pro-inflammatory cytokines like tumor necrosis factor and interleukin-12. Said al. showed that activated monocytes express high levels of PD-1, which might explain the higher expression of PD-1 in CD14 plus CD16 plus monocytes as compared to CD14 plus CD16 monocytes. Triggering monocytes expressed PD-1 by its ligand PDL1 induces IL-10 production, which activates CD4TH2 cells and inhibits CD4TH1 cell function.
Diagnostic use.
A monocyte count is part of a complete blood count and is expressed either as a ratio of monocytes to the total number of white blood cells counted, or by absolute numbers. Both may be useful in determining or refuting a possible diagnosis.
Monocytosis.
Monocytosis is the state of excess monocytes in the peripheral blood. It may be indicative of various disease states. Examples of processes that can increase a monocyte count include chronic inflammation, stress response, Cushing's syndrome, hyperadrenochromatosis, immune-mediated disease, pure granulomatous disease, necrosis, red blood cell regeneration, viral fever, sarcoidosis. A high count of CD14 plus CD16 plus monocytes is found in severe infection, sepsis, and a very low count of these cells is found after therapy with immunosuppressive glucocorticoids.
Monocytopenia.
Monocytopenia is a form of leukopenia associated with a deficiency of monocytes.
Dendritic cells.
Monocytes can be used to generate dendritic cells in vitro by adding cytokines like granulocyte monocyte colony stimulating factor, GM-CSF, and IL-4.
Blood content.